Swayam Prabha. Digital India. Educated India.
So, in the last class we were talking about cell source, right. So, we talked about where the cells will be obtained and what type of cells we can get and so on, because when you are talking about tissue engineering, cells are the second aspect of the triad. And during that time I also said the cells which we get would also have to be cultured. So, there are different aspects to it. So, we identified what could be the source and what type of cells we could use, but we should also know how to actually separate these cells, how to actually get these cells from the tissues, and how to get the specific cell type from the tissue, because the tissue is going to have multiple cell types. So, we need to be able to isolate the specific cell type which we are interested in. So, that is one, also crucial, and once you have that, we should also look at how the cells would be cultured, and in case we are working with stem cells, how the cells would be differentiated, right. So, all these aspects are crucial when you are talking about cells. So, the cell culture aspect is what we are going to be talking about. So, we will talk about first harvesting the cells, selecting and isolating the cells, expanding or culturing the cells, and differentiating the cells. So, these are the different aspects of cell culture. So, harvesting just means that taking cells from a tissue. So, it could be from different tissues. So, you just take out the tissue of interest, and there could just be some simple mechanical digestion or some simple treatment to get a mixture of cells from the harvested tissue. That could be used as the cells itself, that mixture could be used as the cells itself. So, one example would be just using platelet rich plasma or platelet poor plasma, where not much of selection or anything is done. There is a little bit of isolation being done, but it is primarily just harvesting and using it. You could also have selection which could be a lot more specific, where you try to identify specific cell types which you are interested in, and taking those cells and actually using them for tissue engineering applications. It could either be in vitro, or it could be directly injected into a person for a regeneration of the tissue, right. So, these are two options when you are talking about cell selection and harvesting, but the cells you get is usually not of enough numbers. We did talk about this earlier, right. So, we need a lot of cells, and especially if you are going to take it from an autologous source, then you are not going to have enough number of cells. So, you would have to culture them. So, usually this is done in vitro, so that you can get enough numbers. So, even if you are going to harvest 10 power 4 or 10 power 5 cells, you might need 10 power 8 or 10 power 10 cells. So, you might have to multiply these cells over a period of time to get enough cells, and then use it for regeneration or tissue engineering. So, culturing the cells is a crucial aspect, and this is going to be dependent on the type of cell you are working with, right. So, if you are going to use for example, if you are working with hepatocytes, the culture conditions would be different compared to what you would have for an epithelial cell, right. So, you need to know what culture conditions are and how you actually maintain these culture conditions, what are the growth media required and so on. So, if you are using stem cells, then obviously the expansion would also have to make sure that the stemness is maintained. So, in the sense that as it is dividing and multiplying, it should not differentiate, it should only multiply. So, that is a crucial aspect. If it starts multiplying, you will get enough numbers, then the stem cell has to be differentiated to get the specific cell type for the application. So, the differentiation process itself will be different. So, how you direct the differentiation to get the desired cell type is also a challenge that needs to be understood, and this will be different for different cell types. So, first thing is harvesting the cells. There are different methods and procedures. So, the method you use would depend on the cell source or the cell type itself. So, depending on the tissue which you are going to extract, you are going to use different techniques, right. So, most of these cell harvesting techniques actually come from some type of a diagnostic procedure. So, there are different diagnostic procedures for studying for checking the pathology, checking some disease condition. So, here tissues are taken for these studies. So, these techniques are used for cell harvesting as well. So, there is a high demand for patient safety because these procedures actually expose the patient towards potential infections and so on. So, you need to make sure that these are done in proper clinical environment and you do not cause any complication to the patient. So, you need to maintain the correct environment, the clean environment for doing this. So, special care for handling, storage and transportation of these tissues is also required, because depending on the tissue type you might have to store it under different conditions, right. So, you might have to store it in liquid nitrogen, or you might have to treat it as soon as you process it immediately and so on. So, depending, or you might have to heparinize it if you are going to be using blood, or citrated blood might be required. So, depending on what tissue you are working with and how you are going to use it in the future, you would have to handle it and store it appropriately. So, one of the most common sources for cells in tissue engineering is bone marrow aspirates, because these contain the mesenchymal stem cells. So, these are usually harvested under local anesthesia. People are given local anesthesia and it is taken, the bone marrow aspirates are taken from the upper part of the hip, which is the iliac crest. I mentioned this earlier as well. So, this is the iliac crest. So, you have the hip bone. So, what you see here, these are the iliac crests. So, here the bone is the bone contains the bone marrow, and you can actually get a bone marrow aspirate from which you can get this mesenchymal stem cells. These can also be isolated from the sternum. Sternum is what is in your rib cage. So, the rib cage is actually connected by something called a sternum. So, that is what this tissue is, this bone is, and tibia is other bone in your leg. So, you actually can harvest stem cells, mesenchymal stem cells and bone marrow aspirates from these tissues. Okay. So, iliac crest is the most common tissue from where you try to harvest it. So, what you do is, there is something called an aspirate needle. So, these are large needles, maybe 16 gauge or something, and these are penetrated into the skin and to the and it also penetrates the outer part of the bone, which is the hard bone, and it reaches the softer central part which contains the bone marrow. So, this is a painful procedure, right. So, that is why you have to have a local anesthesia. So, you are actually causing a puncture through the skin, through the muscle tissue into the bone. So, it is going to be quite painful, and from this softer part of this bone where the bone marrow is present, the marrow is sucked out using a heparinized syringe. Okay. So, why do you think we use heparinized syringe? What is the role of heparin?
To prevent blood clotting.
Yeah. So, these aspirates can actually clot very rapidly. So, to prevent that, you need to use a heparinized syringe. Okay. So, these aspirates will contain hematopoietic cells, adipocytes, endothelial progenitor cells and osteoprogenitor cells as well. So, if more than 2 ml is aspirated, then peripheral blood can actually dilute the mesenchymal stem cells. So, it is important to control how much you are aspirating, and then whatever you are getting is now a mixture of cells, and you might have to separate cells to get the specific cell which you are looking for. So, another technique which is used is tissue biopsies. So, tissues from almost any organ can be biopsied. So, you basically just take a piece of the tissue, cut a piece of the tissue and use it for your cell isolation. So, there are different techniques for this. A scrape is basically cells are removed from the surface of a tissue, where you just take a scrape of the tissue, or you can have something called a punch biopsy, where a punch which is round shaped is used to cut and remove a disc of tissue. So, this is like what you would see in a punch hole, right. So, you create a small hole, a small punch, and this disc shaped tissue can be harvested, and that can be processed further to get the desired desired cells. You can also have needle biopsy. So, here a needle is used to remove a sample. So, usually this is for liquid samples. So, it could be for the limb, for the blood and so on. You have stereotactic biopsy. Here a stereotactic system which uses the 3D coordinates is used to identify a small target region and specifically remove tissues. So, these things are used in places where you need to have a lot more control. For example, if you are going to take brain tissue and so on. So, you would not want to damage the organ. So, you need to have precise control over which part of the tissue you are actually harvesting. So, for that reason you actually use something like stereotactic biopsy. Yeah.
So, what are reasons for harvesting brain tissues?
You might want to work with brain cells, right. So, there could also be reasons like diagnostics for tumor and so on, right. So, there are different reasons where you do biopsies. A colposcopic biopsy is where a colposcope is used. This is a close focus telescope that allows the doctor to actually look at the cervix to look at in detail. So, this is usually done for again diagnosing cancer, cervical cancer. Endoscopic biopsy is where an endoscope is used to collect the sample. An endoscope is basically a long, thin, lighted optical instrument which is used to get deep inside the body, and it can also examine and operate on organs. Okay. So, you might have seen endoscopy done through the nose or through the mouth, those are very commonly done. Okay. So, this is just a long tube which is inserted into your body, and so these are some of the different techniques which are available readily for performing biopsies. So, what I am showing here is the punch biopsy. So, this is one of the simplest things done. Usually this is done for tissues like skin which are easily cut open. So, what you do is under local anesthetic you take this punch tool which is a sharp knife which is round in shape, and the punch is placed over the area which you want to harvest, and it is pushed down and rotated to remove a circular piece of the skin. The skin sample is then lifted with the forceps or a needle, and this can be cut from the tissue before it is further processed. So, biopsies for internal organs however cannot be done with such a simple and crude way. You would need more of a fluoroscopic control. You would either combine it with endoscope or an X-ray machine, so that you actually can keep track of where you are going and what tissue you are removing. So, the biopsies you got or the aspirates you got basically are a mixture of cells, right. So, you might have to process them further before you can use them for tissue engineering applications. Bone marrow aspirates are basically suspensions. So, cell retrieval can actually be done from these suspensions, whereas tissue biopsies have extracellular matrix attached with it, right. The cells are adhered to the ECM and you have actually taken out the tissue. So, now you need to make sure that the ECM is removed before you start working with the cells. Okay. If you are only wanting the cells, you do not need the ECM. So, this is the opposite of decellularization. You need to remove the ECM without actually damaging the cells. So, the first step is usually mechanical. So, what you do is you could either have a vortexing with digestion buffer or just dicing with scalpel and so on. So, those are simple techniques to try and remove as much of the ECM as possible. The disrupted biopsies could then be implanted or further processed, or it could be placed in culture for expansion as well. So, what else do you think you can do for further processing? So, the first step is just mechanical, right. So, after that if you want to remove ECM, what do you think you can do?
Collagenase digest.
Collagenase or something. So, where you actually use some protease, some enzyme which could actually disrupt the ECM and release the cells from these ECM. Okay, so, that would be one way to do this. Trypsin could actually just dislodge the cells from the ECM instead of destroying the ECM. Trypsinization actually makes sure that cells do not adhere to the surface, they just come out of the ECM. Okay, so, there are different techniques which you can use. Okay, so, there can also be calcium dependent molecules being targeted, which are which are used for cell adhesion. So, you can use chelating agents that will actually target these calcium dependent cell adhesion molecules and help in the release of cells from the ECM. So, basically cell retrieval process can be of 2 or 3 different steps. The first step is mechanical homogenization, where you can use sonication, manual pulverization or high speed blending. This is usually done for soft tissues, and enzymatic digestion is done for soft and cartilaginous tissues, where you use things like collagenase, proteinase K and so on. Acid digestion is done for osseous and fibrous tissues. So, instead of using enzymatic digestion, when you have hard tissues, you can use acid digestion, so which is usually done using HCl. So, the harvested cells, the cells which you have harvested actually contain a mixture of cell types, right, because you have not done anything to specifically attract one cell type. Whatever you have done till now, you have just done mechanical isolation or enzymatic digestion, which will just retrieve all the cells that are present in the tissue. This would mean there is a mixture of cells, but now you might want to use only one specific cell type. So, if you want hepatocytes alone, then you would have to make sure that hepatocytes are separated. So, there are many techniques which can be used to enrich these cells. So, how you do this is dependent on 3 major factors. It is based on adhesion properties of the cell, or the morphology of the cell, which would include the density and size of the cell, or it could be antibody binding and so on. So, the physiology of the cells and so on. So, use based on the distinct characteristics which are unique to a cell, you try to separate it from the other cells. Okay. So, the procedure can be either positive selection or negative selection. What I mean by that is, in positive selection, you try to isolate the target cell type from the entire population. So, you might have like 8 or 10 cell types, and you try to target one cell type and take that out of the mixture. Whereas, the other thing is negative selection, where you deplete all other cell types and only the target cells remain. Both approaches are possible. Both of them have their own advantages and disadvantages. In positive cell types, the advantage is there is high purity, because you have targeted one specific cell type. You know for sure only that cell type is going to come. There is going to be a unique property which you identify, and you are going to use that to separate it. So, which means it is going to be highly pure. It is not going to have any issues with purity. Also, the cells which you get can be further processed again and again for purification, right, because you are having only one type of cells, and even if a few other things are present with it, if one of the techniques you have used is not very specific to one cell type and it can actually attract 2 cell types, then what happens is you now again have a mixture and you can still further process it. But the disadvantage is you have actually used some antibody or a labeling agent that will attract the cell type of interest. So, your cell might actually have this attached to it, and that could interfere in the further processing. So, if you are going to culture them, or if you are going to use it in vivo, this could actually be a problem. Yeah, Vasudha.
Sir, have they tried using selective media or something?
Yeah. So, people do use that. So, we will talk about that as well. So, that is also possible. People use selective media to so just like how you would do it for transfected cells, but that is primarily done for transfected cells. Without transfection, the cells might not have that level of selectivity. So, in case of transfection, you can have something like an antibiotic resistance, and then you culture the cells in the presence of the antibiotic. Every other cell will die and only the transfected cell will survive. But if you are talking about cells which are just isolated, it is not very easy to do that. So, negative selection, the problem is it is very complex to design a cocktail which will actually remove everything except the cell you are interested in. Because you need to now target so many different cell types. So, it is actually a difficult process to do it. Because of this, you would end up with something which is of lesser purity. You may not be able to get the highly pure mixture. However, the advantage is the cells will not have any of the antibodies or labeling agents after you do this. Okay, so, both these techniques can be used. You would have to decide on what is of more interest to you, what level of purity, or whether the cell should not have any antibodies attached or so on. So, depending on what you are going to do with the cells and how you are going to process them, you would choose negative or positive selection techniques. So, when you are trying to isolate a cell type from a mixture of cells, you want to identify unique properties of the cell that of that cell type, and try to exploit that. You want to identify something which is specific for that cell type. So, the choice of isolation technique will depend on the cell type itself then. So, what do you think are the parameters that will actually govern the choice of isolation technique? What do you think will affect how the cell isolation is done?
Cell surface markers.
Okay, surface markers is one.
Density.
Cell density, okay.
Size and geometry.
Okay, size and shape.
Charge.
Cell surface charge, okay.
The type of product we finally want.
Okay, what do you mean by that?
Depending on the final, I guess,
You mean the application or the product?
application.
Okay, so what you are going to use the cells for, okay. Anything else? Okay, so
pH resistance. I mean,
Okay.
can destroy cells.
okay, so pH resistance is one specific aspect, but in general what I would say, okay, so depends on the amount of stress which a cell can withstand, right. So, you might have cells which can withstand certain pH or certain temperature or certain shear, and you can only choose techniques which can handle that. So, for example, if you are going to use something like a centrifugation technique, cells which cannot withstand shear cannot be used separated using centrifugation. You are going to lyse too many cells, right. So, those are factors which you have to take into account. So, another thing is ultimately what do you want, how pure and of the cells do you want, or are you focused on yield, because you want to isolate as many cells as possible, right. In some cases yield is more crucial than purity. You would want to ensure all the cells of the type are recovered, but it is okay if the purity is only 80%. So, you might want to use a technique which is suitable for that. So, again negative or positive selection which will depend on the application we are used and any other specific requirement which the application might have. Also you have to look at time and cost, right. So, you cannot have a process which is too time consuming, which is very low throughput and it is going to be very expensive. So, those things will not be the most attractive options. So, you need to figure out things which are viable for the specific application you are looking at. Okay. So, the aspects like surface charge and surface markers would be the cellular characteristics which you are looking to use for isolation. So, surface charge and adhesion properties would be important because these determine the extent of attachment to a surface and can be used to separate adherent cells from suspension cells. So, there are cells which are suspension cells as well. Mammalian cells which can survive in suspension are there. So, these suspension cells can be separated from adherent cells, right.
What are these cells?
Suspension cells
No, mammalian cells which can
survive under suspension. So, blood cells would be one. So, all the immune cells are also they can survive as suspension cells. Cancer cells can actually survive as suspension cells. There are different cell types which can actually, stem cells can actually survive as suspension cells. So, mesenchymal stem cells are anchorage dependent, but there are other adult stem cells which are and progenitor cells which are actually suspension cells. They can actually form some kind of an aggregate and survive as an aggregate. So, we will talk about it. Okay. So, cell size and density is a parameter which can be used for separation. So, sedimentation, filtration or density gradient centrifugation are techniques which can be used for separating cells. So, these have been extensively studied and people use it very commonly. Cell morphology and physiology, basically the shape, histological staining, media selective growth, redox potential and other visual and behavioral properties of the cell can be used for identifying and isolating the cells. People have used many of these techniques for getting specific cell types. So, last is the surface markers. It is the most specific method. So, what you do is there is specific binding of a surface antigen either to the antibodies or to aptamers to selectively capture cells that have a specific surface phenotype. So, you try to attract cells which are specific which are of specific interest to you. So, people also work with combination of these techniques. So, instead of just using one method, people try to use combinations, thereby getting something which is much more pure, right. So, usually combinations would include one of the first three techniques, first three features and the last one. Okay. So, because the first three aspects are a lot cruder compared to the last one, right. The last one where you are trying to use surface markers is very specific and also highly sophisticated compared to the other three, because if you are talking about just surface adhesion, all you are doing is culturing cells, right. So, you are separating adherent cells from non-adherent cells, it is quite simple, and same goes for cell size and density. Filtration is a very simple process, and so is centrifugation. So, all you need is get a suspension of the cells and centrifuge it, and you will be able to get a gradient centrifugation done, and you will be able to separate the cells. So, morphology, again those things are also not too difficult to do, these are reasonably simple to perform. Selective media and things like that are not too difficult to work with, whereas surface markers can actually be quite tricky and quite tedious, because this requires a lot of information about the cell you are interested in, and designing of proper antibodies and antigens, choosing the correct molecules which can actually adhere to the cell you are talking about, and using very sophisticated equipment like cell sorters which can actually use this to separate the cells. So, what people usually do is perform one of the first three options first and then perform the last one. So, that way you do a combination and you get much higher chance of getting much higher purity, right. So, this is very commonly done for getting specific cell types. So, we will go into some of the techniques. So, this is just an overview of all the techniques that are there, and we will talk about individual techniques in greater detail. So, the first technique is the plastic adhesion, which is based on the surface charge or surface adhesion properties. So, this can either be positive or negative selection. The purity is very low, but the yield is quite high, because if you are trying to separate, all you are doing is segregating it as two different classes, adherent and non-adherent cells, right. So, every non-adherent cell is going to be in the suspension, every adherent cell is going to adhere to the surface. So, you are obviously going to get very high yield, but obviously the purity is going to be low, because you are going to get a mixture of adherent cells and a mixture of non-adherent cells. Density gradient centrifugation is based on cell density, and this is a positive selection for separation, and again the purity which you would get would be low, because your centrifugation is not going to make sure it is distinct separations, and again many cells could have similar densities as well. So, you are going to have cell populations in certain regions. So, the purity is going to be low and the yield is going to be quite high, because in that particular region you are going to have cells, all the cells of that particular density. Filtration is based on cell size. It is a positive selection mechanism where you have low purity and high yield. Again, everything which is larger than the filter pore size is going to be retained, and that is going to be a mixture of cells again, right. So, you are not going to be able to get one particular cell type from that. FACS, MACS, aptamer binding are all surface antigen binding techniques which have very high purity, and the yield is actually not very high, yield is low to medium. So, that is why you try to use the first technique before this, so that you make sure you collect all of the cells before you actually perform further purification. Okay. So, if you were to perform the FACS or MACS or something like that, the cell sorter first, then obviously you are not going to have enough cells to work with. So, there can also be selective growth media or culture where you use the physiology of the cells. So, usually this is negative selection, and here the purity could be medium to high depending on what types of cells you are working with and what actually is the mechanism you are trying to use, and yield could again be low to medium. So, LCM is actually a laser based technique which is used for cutting of cells, identifying cells and isolating cells, and that is a morphology based technique, and this again uses positive selection, and the purity is high and the yield is low. With RBC rosetting and immuno LCM, you are actually trying to use combinations here. So, RBC rosetting is the basically using the size and surface antigen, whereas LCM immuno LCM would be using morphology and surface antigen. These will give you very high purity because obviously you are using the surface antigens as the second technique. So, you are going to have very high purity. Yield will again be medium to low. Okay. So, these are the general techniques which have been extensively studied, and we will talk about some of these techniques in greater detail, and some of them we will not discuss. Let us first look at the first criteria, which is adhesion based isolation. So, as I said, there are two types of cells, adherent cells and the suspension cells. Adherent cells basically require a suitable surface on which they can attach in order to thrive. Examples would be macrophages, fibroblasts and mesenchymal stem cells. Suspension cells do not require an attachment surface and can occur in suspension in the body. They are usually found in fluids like blood or lymph, right. So, all your blood cells are suspension cells, right. They do not have to adhere to something to grow, they will survive in suspension. So, lymphocytes, granulocytes and other immune cells are all examples of suspension cells. So, cancer cells which have actually lost the ability to be adherent are also suspension cells. Okay. So, some cancer cells due to the mutations lose their ability to be adherent. So, they do not have to be adherent. So, they will be in suspension. So, those cells are also suspension cells. So, this technique has evolved as the cell culture techniques have evolved. So, when cell culture was first done, people are using just Pyrex glass flasks. So, on which the cells would they are trying to culture the cells, but this does not actually work because primary cells do not attach to glass surfaces. Glass surfaces hydrophilic or hydrophobic? Okay, why do you think it is hydrophobic? Why do you think glass is hydrophobic? Uncoated glass is hydrophilic, it is not hydrophobic. Okay. So, the glasses you see like what you wear or all the window glasses and all, they are coated. So, that is why you see droplets standing there. Otherwise, water spreads very nicely. So, if you were to perform a contact angle study on a pure glass, the contact angle would be close to 0. So, it is a clean surface would be very hydrophilic. Okay. So, but the cells do not like very hydrophilic surfaces as well. They do not like very hydrophobic or very hydrophilic, they like only something in between. So, because of this the primary cells do not adhere to Pyrex glasses, and also Pyrex glasses were actually too painful to maintain, right. So, you have to actually sterilize them, you have to clean them, there is a lot of cost, it will get broken. So, it is very painful to deal with glass culture plates. So, in the 60s disposable polystyrene based culture plates were brought in and people started using them more commonly. They provided the same optical clarity as glass, but they were more malleable and easy to sterilize. So, so these were advantages to use them, but the problem was they could not actually solve the cell adhesion issue because this was highly hydrophobic. So, your polystyrene is highly hydrophobic. So, because of this cells would not adhere. So, then people started looking at surface modification of these cell culture plates, and first thing which they tried was simple oxidation using ozone or sulfuric acid and potassium hydroxide, hydrochloric acid and so on. So, due to this oxidation what would happen is there is a positively charged surface, the cell surface is negatively charged. Because of this, and proteins are negatively charged and so on. So, all these things, because of this they will adhere, and once they start adhering they will start growing. So, that is what was tried out as the first surface modification. So, then people started coating ECM proteins. So, proteins like collagen, fibronectin, chondroitin sulfate have all been coated on these culture plates to make sure that cells will adhere very nicely, and these plates are actually very effective. So, another technique which is being studied is also to use polylysine to provide a positively charged surface. Again, that will help in the negatively charged cells to adhere. So, these techniques have actually improved cell adhesion properties, and now culture plates are very effective for cell adhesion. Cells will adhere very nicely on culture plates and they will grow without much of a problem. Now that people have evolved with the cell culture techniques itself, the adhesion based isolation could also be evolved. So, for example, if you want to isolate macrophages which are adherent cells, what you do is you take either bone marrow or peripheral blood, and the mixture of cells are seeded on these polystyrene plates with serum. So, the monocyte and monocyte or macrophage differentiation cytokine cocktail is also given, that is the media which is supplied for the cells to grow. After 5 to 7 days, the cells will differentiate and form an adherent monolayer, while the unwanted cells remain in the suspension and are discarded. So, whatever bone marrow you had, like the stem cells you had in the bone marrow or the peripheral blood, would have converted to form macrophages because of the differentiation cocktail, the cytokine cocktail, and then you have only the macrophages which are adhered to the surface. Everything else will be in suspension because the others are all blood cells, right. So, they are not going to adhere. So, this way you can get macrophages with reasonable purity. If you are looking at isolating suspension cells, what you do is you culture it in ultra low attachment plates in the absence of serum. So, now these plates could either be very hydrophobic or very hydrophilic, and they will not support cell adhesion at all. So, what happens is the cells which can actually survive as single cell suspensions or aggregates in suspension will survive. Other cells will not have any place to adhere and they will die. So, you will get all the suspension cells only, all the adherent cells would have been killed off. So, this technique is specifically used for isolation of macrophages and adult stem cells and progenitor cells from differentiated cells, and for isolation of cancer stem cells. Okay, so, for these applications people have used cell adhesion based isolation. The advantage is it is a simple procedure which is reproducible and cost effective, but the limitation is the purity of the recovered cells is low, because obviously you are getting a mixture of cells which could adhere or which could be in suspension, and you will get only that. And there is also a risk of cross contamination. Anytime you are actually performing cell culture, there is a risk of contamination, right. Contamination is one of the major challenges when you are talking about cell culture. So, because of this, that could also happen. You could always have some bacterial contamination or fungal contamination which will completely spoil your cultures. So, that is a problem. So, because of these advantages, because of these disadvantages of low purity, people have looked at other techniques which are more specific to if you are looking at getting a specific cell type, right. So, you cannot always purchase cells, right. You would have to isolate cells from isolate primary cells from tissues. So, when you have to do that, you would have to perform different studies.